So today's lecture, I'm going to talk about learning and memory, and especially synaptic learning rules. So the question is, how can you store information in the brain? There are already, I listed like 3 different general types of mechanisms that you can store information. So one is a sustained activity in a recurrent network. You have already seen this, say, in a half-wave network, right? You can use sustained activity to store like a memory pattern. But those actually store as a synaptic connections. But the memory itself manifests as the dynamical stable state. There are also other types of the sustained activity. I'm going to give you examples in a moment. Another method is sustained activity in a single neuron. Because in a network, of course, you need a synapse, right? You need a hold activity. The activity has to go through synaptic transmission. But it's actually possible also in a single neuron without any synapse. So I'm going to spend most of the time talking about plasticity in synaptic connections. What are the synaptic learning rules, right, to compare. We will compare that with different theoretical ideas and actual experimental data. All right. So I'll give you one example of sustained activity. This is in the network. This is a ocular motor. It's where you control eye movement. Some neurons. So this is the eye position, right? When you move your eye to this is a good fit to a new location, it's sustained there, goes sustained there. The activity of the neuron, you see, have a different firing rate. So you plot here, the firing rate is almost like a linear function of the eye position. Right? So this is the instantaneous firing rate. Right? Just like you have a small time window to see the instantaneous firing rate as a function of time. So you see there are different levels of activity, right, that correspond to different eye position. This is called a neural integrator because these neurons don't get information directly from eye position. They got the information from eye velocity, the movement velocity. You have to integrate velocity to compute your position. Right? That's why this is called a neural integrator. So the simplest model is first proposed by David Robinson at Johns Hopkins, actually, developed by other people. So the simplest idea is that suppose you have a single neuron. Actually, you have a group of neurons. You have a synapse onto yourself. All right. So if you write an equation, it will look like this. It's a linear decay term. There's a connection weight W. The gain function is that you translate this, like let's say this kind of a threshold linear function. You translate, it transforms the voltage to firing rate. All right. The I will be external inputs. So in a special case, because if you don't have a connection, this system will just decay exponentially. Right? It's not very interesting. But if you have the connection weight W, if it's equal to 1 exactly, then because the gain function is linear whenever your activity is greater than 0, so for positive activity, so this thing, you write like this, right? This thing goes away. So what you got is this activity is a function, derivative is function equal to the input from outside, right? So then the solution, of course, is integration. That this voltage reflects the integration of the inputs in time, right? So you get something like this. This is the simplest neural integrator model. It's based on connections which are precisely tuned, right? To do this integration. So this is standard model, and also there are a lot of evidence for this kind of computation. Right? This is really integration in the sense of calculus, the Newton-Leibniz formula. This is exactly what we show here. So another type is a single neuron. I just only mention this experimental data because I thought this quite eye-opening. Right? This is a single neuron. You use some all the synaptic connection actually blocked. It's a single neuron in a slice. So when they show how you have a stimulus, there's a current injected into the cell. All right, this is the spikes of that neuron. Of course, when you inject current, this is the instantaneous firing rate, right? When you inject current, of course, the cell fire a lot. But look at here, when the current is removed, this is a very long time scale. This is let me see how many, 5 seconds or 7 seconds, right? It's very long. You excite the cell for a while, the fire is high, but when the stimulus is removed, over here there's nothing, right? It's the same as nothing happened before, but you see the sustained activity actually higher, okay, sustained activity. If you do that again, it goes up again, all right? So the sustained part of the signal is sort of like an integration we just saw before, but this is a single neuron, all right? And this can last many minutes, right? Some of the cells can last half an hour, last for a very long time. And if you inject current in the opposite direction, the fire rate can go down, right? It's bidirectional. It's really pretty amazing. This is the first found in the entorhinal cortex. It's where the location you found those gray cells. Remember the gray cell, the periodical. That's the same brain area. It's very strange. So these neurons, this is sort of like an integrator, just we showed before. But instead of using synapse, use a single neuron. And how this works, it's not exactly sure, but it depends on the calcium current. Calcium flowing to the cell. Presumably, the calcium open up some channel, like you have sodium flows in constantly to drive the cell. So this cell looks like it's have a continuum of stable states. Right? Okay. If you make a cell it's bistable, either on or off, that's easy, right? This is harder to do something like that. It's just totally amazing. Now, this cell turns out, it's not just in the entorhinal cortex, it's in the prefrontal cortex, which for short-term memory, which for planning, for those kind of stuff, also even in sensory cortex. So this will, really change the rule of the game. So in the past, when you do something like this, you have to think this must be synaptic effect, it must be a network effect. You see, the single neuron can do something pretty unexpected, actually. All right, I'll just give you the example. All right, now we'll switch gear. We'll focus on this synaptic plasticity. There's some explanation. There are several classifications. One called supervised learning. So we will learn this for the simple perceptron. Right? The learning is that you have some inputs, you have some parameter you can change, like a synaptic weight. You want your output to be closer to some desired output. Right? This is called a supervised learning. Is that there's a teacher who knows the true answer, right? What your output should be, and then you can update your system to update the synaptic weights and the threshold to learn the system. So this is perceptron, a deep network based on the same kind of idea, just had many more layers. Right? This is called supervised learning. This is to compare with they call unsupervised learning. There's no explicit teaching signal. So this would be like in the Hopfield network, we learn by sort of Hab rule we talked about before, right? In that case, we don't have a teacher tell you what the output should be. You just change it. Once you have the inputs, you just multiply them, you just change the weight. That's it. It's called Hab rule. There are also other methods I'm going to talk about very briefly, independent component analysis, and also their self-organizing map, which I will talk in greater detail later. All right. So there's another type of learning, it's in between. It's sort of like a supervised, but not completely. So this is the sort of, you maximize some reward in a stochastic environment. So the teacher doesn't tell you what your output should be at every step. It just but it just gets awarded at some time. So you do have like a credit assignment problem. Sometimes you get something good, you're not really sure what you did in the past actually eventually lead to that reward, right? It's hard to assign the credit. So this example, I'm giving a very brief example, time-different learning, and all these examples, three, have not only application in, say, like machine learning, and also has application in the neural system as well. Okay, I'll give it a very brief explanation. We start with supervised learning. You have the perception. I'm not going to repeat. When you look at the learning rule, it looks like there's a product of two terms, right? Two factors. One is the input, is x. So another factor is something to do with output, right? It's output times the input. The output here is a difference between the actual output and the desired output. This is based on that gradient descent of square error. So what's the example? Where do you see this kind of learning? This example, I think one example is a cerebellum. Right, cerebellum, I'm sure you already learned this example before, right? In other lectures, is that you have a prism, so your visual target is displaced. You throw a dart, you have error, right? But you quickly will adapt. But once you remove that prism, it goes to the other direction. So where does learning take place for this kind of task? I think presumably it's in the cerebellum. Right? Cerebellum is a small brain. It's good for this kind of task. In the cerebellum, the learning rule in the first model is very similar to this kind of learning rule. Right? It's like this. So the learning happens at the synapse from parallel fiber to the Purkinje cell. Purkinje cell, as you know, is the only output of the cerebellum. Cerebellum has lots of other cells, but only one kind of output. So these cells, the synapse, a lot of parallel fibers, like this parallel onto the cell. The cell has like a fan-like shape, it's very strange structure. So the teacher here is called a climbing fiber, which signals error, right? And the student is a parallel fiber. So when the teacher fires, or there's some error, the desired output and actual output is actually different, right? And then you change the synapse from a parallel fiber to the Purkinje cell. Right? So I'm not going to go into detail of this, but I just mentioned that this is the evidence that this thing actually exists, right, in the brain. I give you another example. This is also the prism experiment, but this is for the binaural. They have very good hearing, right? They can, based on hearing, they know the location of an animal, of a rat, even beneath some snow, right? In this case, the evidence is from optic tactum, it's basically superior clictus, which will control eye movement. But in bird, this is the highest visual area. They don't really have a cortex. So some of the neurons are multisensory, so they have an auditory receptive field and also visual receptive. They can. So normally, so basically in the field here, in actual space, right? For this particular cell, if you shine a light in this location, it drives the cell. Or you have an auditory target, a sound, the broadband sound coming from this location, cell also excited. So the visual receptive and auditory, normally they are aligned, all right? So if you have the prism like this, of course the visual receptor is displaced. Auditory receptor is the same because your ears are not changing. Only the visual receptor is changed, right? In this case, actually, the visual receptor actually is wrong, right? And that's not because you have this prism. So after a few weeks, you see they're aligned again. The question is, which one is a teacher? Which one is a? It turns out the auditory, the visual signal is a teacher, right? Because it doesn't change, right? So but auditory receptive field is moved, shifted to be aligned with the visual receptive field. Right? This is another example of the supervised learning, right? All right. So this is just like a very general overview of the supervised learning. Now I'll give a very quick example of reinforcement learning. So in this case, well, I don't have time to give a thorough explanation. I just very briefly, I just pick up like a one feature which is very relevant to our argument. So the framework here is that you're in a random environment. So you want to predict your reward based on some sensory inputs, right? You try to predict your reward. So your reward at time t, it depends on your sensory inputs. Let me see. The sensory inputs is x. All right. So x at the time t and t minus 2 is in the past. Okay. You have different weights. You have to weight your sensory experience in the past, try to predict the reward right now, right? Any good sign right now. If you want to say that's the capital R, this is the actual reward at time t. You want to minimize this function, right? This R, small r, is your predicted reward, or your predicted reward based on your sensory experience in the past, right? And you compare with your actual reward. If you want to minimize the square error, try to find the weight in the proper way to how to weight your past experience to predict the future reward. And based on gradient descent of square area, you get a learning rule look like this, right? Just very similar to the perceptron, right? It is something related to the output here, just reward and your input in the past, right? The weights actually have a index as a different time in the past. So basically, you see this also, a product of two terms, and this term, the reward, big R minus R, is reward prediction error, right? Is your actual reward compared with your predicted error. And this is a sort of one key feature of this kind of learning, right? So time difference learning. This is not a full coverage of the entire topic. I'm just picking this out because there is amazing biological data for this piece. This method is used for artificial intelligence. I think many decades ago, the first use in backgammon. Right? It's a stochastic game which is already played better than human expert. In, I guess 20 years ago, it's already better than human. And AlphaGo is a more recent achievement. Right? This method is based on. But it's used the reinforcement or learning in the actual setup of the system. Right? They also use deep learning, like multi-layer perceptron type of learning as well. So it's a very complicated system, but it really uses biological-based learning inspired by biology. And this Go game is presumably the hardest human board game. Right? This is just last year, I think, the first time. Actually, 20 years ago, a computer is already better than human master in chess play, right? And the Go was better than human just like last year. So now the human is much worse than the system because the computer getting better after a few months. The human not getting better that fast. So now I think there's no board game like this. Human is better than computer, right? So, and the thing that actually works is based on the neural network model is used as a part of this system. So the biological evidence for this, I think this is amazing. This is the first time the biological have that prediction from the reinforcement learning theory. And in the brain, they find some signal in the dopamine system. You know that the dopamine is a system that's important for reward, right? So that's why a lot of people take drugs, because they can get this reward dopamine without actually doing anything good, right? So for the dopamine system is that typically if you have a reward, let's say here, so for animal it would be like some juice, some food, human maybe money or something. These cells, the far end of the cell, have a big increase. Right? For the real reward, right? For the natural reward, the cell far rate will increase temporarily. So if you do this conditioning test, like a Pavlovian conditioning, you hear beep, right? Eventually you get a reward, right? So after some training, eventually you see the dopamine neurons start to respond to the conditional stimulus, that beep, right? It doesn't respond to actual reward anymore, because now you're able to use the sensory input, that beep, to predict what you're going to get over here, right? After some learning. Now you see your predict reward and actual reward is actually the same, right? So there's no response. So the dopamine neuron response is not just reward; it's predicted reward error. Right? Is your actual reward above your prediction? If you get the same, if you predict the same thing and actually get the same thing, the cell is not active. Right? And here, let's say, if over here you have a beep, but there's no reward, you see the activity actually dips down, right? It's a negative response. So really, this neuron really signals, it really seems to very similar to what you would expect for prediction error. The response of a neuron is the actual reward minus the predicted reward. All right? This is one evidence for that kind of reinforcement learning. All right. All right, this is very brief coverage of the topic. Then now we switch gear and talk about this unsupervised learning. We talked about this Hab rule before as a product of two terms, the input and output. Right? What's the evidence for this? Well, so one thing very similar is called the long-term potentiation. You have already learned long-term potentiation in your synaptic learning lecture already, right? Have you learned already? No? Yes. Yes. Right. So I'm not going to give you a more detailed explanation. Except for I want to ask for one thing, is that for the Haber rule, you see, you strengthen the synapse, right? The condition you strengthen synapse is that the two cells should be active at the same time, right? So it's reflected in the product term. It's like a logical AND, right? A and B. So if one of them is 0, there's no activity. This thing doesn't work, right? So both has to be active. So for this learning, like long-term potentiation, so what do you mean by both active? So you have some synapse from other neurons, right? You stimulate the synapse, and there's this neuron. So if you based on that Haber's idea, you would say that this synapse is strengthened only if the presynaptic cell is active, meaning these fibers active, and this cell itself is also active, right? So if your stimulus is too weak here, the cell here is not active because it's too weak. So if you have very strong stimulus, called tetanus, right? It's very, very strong, so that drives the cell active. Then the synapse get activated by only a few minutes, right? It's getting activated very quickly. So the mechanism for this, how is Haber implemented? Is that there's special, you remember this, how the synapse work? I just look just very briefly. So all the things implemented by this NMDA channel. NMDA channel is kind of special because ion channel are usually opened by either some chemical or some, like a glutamine, right, some molecule, or they're opened by, say, like a voltage, like a sodium channel, potassium channel. So NMDA channel is interesting. It's open only by two factors have to go together. So that's the implemented Haber. It has to be both. So one factor is about the presynaptic cell, another is about postsynaptic cell. So the NMDA channel, where is this one? This NMDA channel, it's activated only if you have a glutamine, right? And the cell membrane is depolarized. What does this mean? Glutamine means the cell, okay, this is the pre, this is the post, right? Signal goes from up to goes down here, right? The release of glutamine means that the cell, the presynaptic cell must be activated, right? At activation, then there you release glutamine. That's one condition, presynaptic cell has to be activated. And the depolarization means this cell in the postsynaptic cell must be activated as well, right? If it has to be depolarized, that means this cell has to be activated, not just from this, maybe from also other channel at the same time, right? So once this channel is activated, a lot of things may happen. The total effect makes the synapse stronger, right? You remember? So yeah, and the key condition is that how this is related to that Haber rule, because the opening of the channel really need activation of both presynaptic and postsynaptic cells, right? All right. Simple Haber rule, mathematics. Yeah, this is an optional material. I just want to mention a little bit so you get some idea. For this simple rule, what does this actually does mathematically? All right. This is very simple, right? The weight vector times their input vector. There's multiple input, multiple weight, right? If you change the weight proportional to the product of their output, here the scalar, and your input, which is a vector, right? If you do this, okay, that's equivalent to this. If you learn this over some time, right, you have lots of inputs, it's randomly varying. After a while, you accumulate your weights according to this. This is the same as this, right? After a while, what do you get? It turns out the weights, of course, based on this, the weights will keep on increasing, getting bigger and bigger, right? There must be some way it has to go down. There's some limit, it cannot go further. But it will keep on increasing. So in the hippocampus, actually, this thing actually happens, right? You know the seizure or epilepsis is often start in the hippocampus because hippocampus has the most concentration of NMDA channel. That will increase the Haber rule. But once you have a lot of cells active at the same time, they tend to be strengthen each other and the waves even stronger. You got a positive feedback. Eventually, you get run out activation. That's a seizure. So the system also had a lot of inhibitory cells. There are only one kind of excitatory cells. There are many kind of inhibitory cells to regulate the system. Right? It's a very sophisticated system. Otherwise, system will go away and get into seizure. So by the Haber rule, you see it tend to the length with the normal vector weight will tend to increase indefinitely. Right? So another thing that's very interesting, maybe you didn't expect, if you don't think about this, is that the directional vector approaches the first principal components of your input. So you all learn, I think the principal component analysis is used a lot, pretty much everywhere, right? So simply like this simple Haber rule, it tend to pick out the first principal components. So principal knows that in two-dimensional space, x1, x2, right, two different inputs. Suppose the distribution of input is a multivariant Gaussian, all right? Let's assume that. In that case, the principal components, the first principal component will be in the direction where you have the most variance. So that you have a covariance, it's an ellipsoid, you have the longest axis, right? It's your first principal component. And their second and third, they are all orthogonal, right? They're pointing to different axes of the equal covariance ellipsoid, right? So there's one clever thing I want to mention. So you have the learning rule like this. So this is a continuous version of the same learning rule. You have a product, output, input. You know that this will tend to grow indefinitely forever, right? So how about I add some like a weight dampening, right? If you minus the weights, like here, this weight would decay term, right? Weighted by a square of your output. Why square? Because this is very clever. If you do this, the norm of your weight vector is going to be normalized automatically, all right? And your weights become to the first principal component of input exactly, right? That's very simple. So you basically have this is a modified Haber rule called algebra learning rule, kind of a very clever trick, right? It's just the Haber rule, you minus the decay. If your decay rate is clever, then this thing will be exactly your first principal components. All right. Well, I'm not going to prove this, all right? So what's the relevance? Is that the brain actually does the principal components analysis? Well, in the past, people suspect maybe you have a hierarchical organization, say your visual system, you have a different receptive field, you go high up, the more and complex, maybe there's sort of principal components analysis, at least in the beginning. It turns out not exactly right. Let me show you quickly this story. So this is a visual receptive field in V1, right? It has this bar, it likes some orientation. The standard model is called Gabor filter, which is basically it's a two-dimensional Gaussian times a straight like a sinusoid, right? It's a sinusoidal wave, like a plane wave, right? Then you get something like this. So it's kind of fit the data pretty well. I think the simple cell in V1 looks like a Gabor filter. That's the first approximation. So people try to do principal components of natural images. So basically, you have a patches image. Each pixel is this pixel you take from a computer screen of a picture. So that's one input vector. You take a bunch of patches from different pictures. You do a principal components. What do you get? Right? So that would be maybe the receptive field of some linear neuron. So this is a typical result. Look like this. This is the first principal components. It's just a constant, which is like an overall level of it's average like a luminance, right, of the patches. The second one looks like oriented. People say, oh sure, this is like oriented filters, right? It's like maybe like V1 cells. But you go to higher order, you see there like a checkerboard pattern, higher. So like a Fourier analysis of different frequency. The problem with this is that receptors are too big, right? No matter how big is your patches, it fills the patches. The real receptor field actually is zero everywhere. It's very compact, very small. A better way to do it is like this. This could use the principle, it could independent component analysis of natural image. You do the same thing, you get things like this. Really look at Gabor filter. And the most part of the thing is zero. It's only when you sum small part, you have these edges. It's pretty amazing, right? So what is this principal components? So this one, to explain it, is kind of hard to explain if I go through all the mathematics, because this lecture is not enough. I'll just give you a very brief explanation. It's related to the blind source separation. So the idea is this. Suppose you have a bunch of sources of signal. Let's say here it's 3 different musical instruments. Suppose you have a 3 recorder over here, so like a recording, like microphones, right? And each microphone is a linear summation, a weighted sum from all 3 different sources, right, with a different weights depending on location. So in all the 3 recordings, you will get a mixed signal from all 3 sources, right? So the question is this: suppose I give you 3 recordings, the linear combination of 3 independent sources. Can you go backwards? Can you recover all the 3 original signals? That's a question of the blind source separation. Right? I don't know how you combine them. I have no idea. All I have are just 3 recordings. This is kind of very useful, right? If you can do that, you can separate the vocal from instrument, right? For separate different instruments, and you can mix them differently. That'd be pretty cool if you can do that. Is this possible? It turns out this is actually possible. This is possible because the signals from these are not Gaussian. So even these were Gaussian, the linear combination is still Gaussian. You cannot do it backwards. But in reality, they are not Gaussian. Both visual signal, auditory signal, they are not Gaussian at all. They have a longer tail than you expected, right? And this can be done. I'm not going to show how this is done, but you just want to recover the sources as independent to each other as possible, or as non-Gaussian as possible. When you mix together, they're closer to a Gaussian. You go backwards, it's less like a Gaussian. All right. I'm not going to, but just say that if you use this method instead of the simple Hab rule, you get a Gabor filter to look like a real thing. And you can also do this for the auditory system. What if you do for natural sound, you apply the same method, you get a filter more like a gamma tone filter, very kind of similar to what you get in auditory nerve fibers, right? All right, so in the next 20 minutes or so, I'm going to talk about another, more detail about another unsupervised learning. So that independent component analysis is also unsupervised learning, right? There are no teacher, they didn't tell you anything, you just have an input. You figure out what's going on, right? So another type is, has some spatial information called self-organizing maps. I'll give you a little bit more detail. So let's take the example, all right? This visual system, we know the visual field is mapped to the visual cortex. There's it's called topographic map. Right? This in the visual field, these lines are mapped on the piece of cortex like this. You can see the shapes. This is actually use activity of these flickering black and white stuff, and based on where you take up that glucose, you got these kind of forms. Right? So this mapping is it's sort of like a conformal mapping. It's like a log and complex domain, log map. So basically half the visual field, you map to some shapes that look like that. Right? It's like a distorted. So there's one interesting based on this idea, is that people have the hallucination induced by drugs. Right? Some of the geometric pattern like these, right? That has an interesting interpretation, that if you do this mapping backwards, right, if the visual field looks like this, where it's a radial pattern versus a spiral pattern, so what this shape looks like in the visual cortex, right? It turns out to look like it's either this one will look like a vertical stripe, this one more like a oblique stripe. This is a two-sided cortex, all right? So it means that if that drugs, right, introduce like a plane wave on your visual cortex, you run in parallel, either way, the other way. So then in your visual field, it would as if you see these kind of geometric patterns. Right? So this is just like one possible link between that particular shape of map and some activity in your cortex. All right, I thought this is kind of a very neat stuff I want to mention to you. All right. The visual cortex. Let's get a little bit more. So the visual cortex, half of the macaque brain is actually visual cortex. There are many different parts doing different things. So I want to say that there are multiple, this means there are multiple topographic maps. So this is based on fMRI in the human cortex. So that's not just a monkey. The human brain is kind of similar. So you use this kind of activity, the visual stimulus look like this, right? You see which part of the cortex is active. This is like in the radial coordination, which is the angle. And you do another one, is this, like, I didn't show this, like it's an expanding ring. It's like, based on these two, you see the activity in multiple places. Then you can figure out actually there are multiple maps, right? There's like a V1 like this, based on topographic, right? You know the location, you know which cortex mapped to which part of the visual field. Then you figure out there are multiple visual cortices. It looks very similar to a macaque monkey. You have a V1, V3, V4, the MT, MSC, so on. Very, very similar. All right. So now I want to mention to you about this one theoretical idea related to the map. It's called the self-organizing map. That's unsupervised learning. You just give them the input organized by itself to get these different maps. So for visual cortex like a V1, there are multiple things mapped on the cortex, right? You see that it's very hard to, you know, it's actually impossible to have a continuous map of everything on the cortex. The cortex is physical, it's a two-dimensional sheet, right? And you have multiple variables. So like here, this is first of all, you have a retinal top is XY position. Where is XY position on the visual field? You already say there is already a topographic map there. On top of that, on the same spatial location, there are other things like the orientation. Right? There are different orientation. For example, the cells here, the different color means the cell here like this orientation. Right? Cells, the green one looks vertical. But all the cells here, they all like the same physical retinal topical location. But all on the location they also like other things, like the other feature, like orientation. And also the same map superimposed on this, they have a ocular dominant column. And there's some cell like the left eye, some cell like the right eye, some cell like both, right? So there are multiple features mapped in the cortex. But I think one take-home message is that the cell in the neighborhood, they close to each other, tend to like similar kind of things, right? And because it's not possible to map everything together in a continuous manner, there are singularities. There are fractures, there are defects. It's just mathematically impossible to map everything continuously. If you go to higher level like cortex, like visual cortex, you see similar things. So this is the surface of a cortex. It shows schematically what the thing the cells are like. They have like a different kind of shapes. They cluster in different blobs. And there's here the sort of cell like faces in different poles, right? So this is actually recording on the surface cortex. The black area means activity. You change the head at different angles. You see the activity spot shown here is moved continuously. That means you really have a map of different poles of the head on the surface of the cortex, right? It's the same thing. Yeah, of course you know the somatosensory cortex. This is also similar to visual cortex. They have multiple maps. The same is true for auditory cortex. The same thing mapped multiple times. In different parts of the body is mapped on the cortex. So this is this stripe, right? This stripe in the brain. So this is kind of like this example. Like this is for a mouse, right? A rat. So their whiskers are very important for them. So this is their body's mapped. This is some of the sensory cortex. You see their whiskers overrepresented, occupy a big area. Like a human, the fingertip occupy a big area, right? And you can see these columns which correspond to each individual whiskers. The cluster of cells correspond to each individual. There's 1, 2, 3. It's pretty detailed map. They're right there. All right. So these are just show this example there. These maps are very general. It's in the cortex. It happens in many, many places. All right. So what is the theoretical idea behind this stuff? So Kahana has the idea of a self-organizing map. So this is not, I won't say it's biologically realistic, but it captures something about the system, right? It's kind of interesting. People use it. It's very famous. And I'll just give some explanation of these maps, okay? So the idea goes like this. Suppose you have a two-dimensional sheet of cells, all right? Let's represent the cortex. You have a grid, IJ. IJ means location on that sheet, all right? So you assume here that all the cells on the sheet receive the exact same input in the beginning, all right? The difference is just the weights. The weight is a vector. The input is, in general is a vector, all right? You have input at the location R. R is that location, the grid, all right? The weights may change from cell to cell, all right, from location to location, but they all receive the same inputs. So the goal here is just say we start with some random connection. Everybody is wired up to the same input, potentially, but because of learning, that's unsupervised learning, I don't tell you where you should go, what you should do. Just based on some learning rule, sort of like a hybrid, I just explained to you. you will change the weight, such that after some learning, you automatically generate a map in the sense that the cell in the neighboring area will like similar things. All right. So the rule is kind of simple. It works like this. So the learning rule, the weights, right, the proportional to a product of two factors once again. So the one is sort of related to the output. So another is related to the input. All right. So the output part is called the neighborhood function. What you did is that if you start from random initial condition, initial weight, you gave it any inputs. Each of the cells will respond to some extent according to this dot product, right? Some cell may happen to fire more actively more than others. So the most active one, I'm going to draw here, we call this it's a winner, right? Its location devoted by R star. That's the winner, all right? Given that the winner, you assume you want to update all the neurons in the neighborhood of the winner, all right? It's just depending on the location of your neuron. The force of here is a Gaussian distribution. Right? If it is a maximum if you were at the winner, if you move away from winner, this term is going smaller. So this term is basically somehow related to the output. It's a winner. If you're close to the winner, you get a big number here. That's it. If you're very far away, it's 0. You don't change the weights. All right? You only change all the weights close to the winner. All right? So the other things depend on the inputs. Right? The inputs minus the weights. So this is not exactly input minus weight. So here is the argument. So the difference, all right, is that if you have two vectors, the input, another vector is output, let's suppose they are normalized, it's only the direction matters, I assume that, then their difference, the difference of the two vectors is the smallest when the overlap is the biggest, right? Because of this, right? The square norm, you multiply them, it becomes this is a w squared x squared times the cross product term, right? So you have these, the difference between vectors is the smallest when you have a big overlap here, right? This is the product is the highest. So let's try to justify why you see this term, right? This term is really similar to this product, except it's a negative, right? All right. So how does this system work? So this gives you shows the process. Okay, basically you have a bunch of neurons. Here, suppose each neuron has a weight. This is like a two-dimensional. You have a vector pointing to some direction. That's the weights of that neuron. All right. And suppose you have an input somewhere. Let me see. Yeah, this small arrow. This is the inputs go to every neuron. It's the same input everywhere, right? So this is two-dimensional. Suppose the input is shown at this vector, all right, pointing this direction. So now you see any of the vector that matches this one will give a big response, right? The weight and your inputs, they match each other, and this one will happen to be this one. And then the neighborhood function. This is the winner. Then you update all the cells close to the winner, all right? And then you have input in a different direction. This one happens to be winner. You update the cell in the neighborhood, right, according to the learning rule, and you do that again and again, so on. So what do you get? Eventually, you develop. But this is kind of shows the weights. Initially, you have a small weights all clustered here, but after learning, you generate a two-dimensional map, right? But if you do this stuff, is that you if you have a two-dimensional input, try to map into one-dimensional network, you get this kind of like a piano curve. Try to get space filling curve. Try to fill in the curve like that. All right. All right. This is a, and this one tend to be. I think that's the last one I want to show you. All right. Oops. You can use to map some more abstract spaces, right, in the cortex. So like say in this case, suppose you have a different animals. They have different features. Like the dove, the hen, they're whether small, big, or larger, either or one. They have 4 legs, 1 leg. You have hairs, hooves, mane, so on. They like to hunt, run, fly. So there's some feature of the animal. So each of the animal has some feature. Like a dog, it's small, it's 2 legs, it has feathers, it can fly, it can swim, right? And so on. The different animal has this. There are also an additional line of input which identify each of the animals. So for each animal, have an extra line. Okay, these are just a bunch of input, high-dimensional input to the system. You have a 2-dimensional grid, different cells, random weights, and you learn all these animals, right? Get input. You don't do anything, right? Eventually, this is the after system. You learn for a while, you get. Then you can test each neuron, can see what the neuron likes. This neuron likes dove, this neuron responds more to goose, and you get some map, right? See all the neuron here is kind of flying animals, right? And the eagle and the hawks are here. These are some of the hoofed animals. Some these are like a predators, right? So it means the input with similar kind of feature tend to cluster together. So this is the. This is just want to show this general method will work for arbitrary kind of inputs. You can for high-dimensional input, you can cluster them together into different different categories. So okay. So I think I also give you a homework today. It's using this self-organizing maps. You can play with it to see how it works. All right. So any questions about this lecture? Okay. Okay, that's all for today. Thank you.